Hello guys, welcome to Dr. Science. So today we are going to talk about what is meant by porphyria. So as you can see here, there is a diagram which is given you in your biochemistry textbooks, and it is quite complex to understand this one, but I will try to make it as simple as possible. And not only that, I will talk about RBC structure in order to understand this one, and after that I will try to explain how the protoporphyrin ring synthesis take place in our body. So this is your synthesis of protoporphyrin ring. So this entire sequence I am going to tell you guys. Okay, so first I will start with the basics. So as you already know, this is your red blood cell, and inside red blood cell we have a lot of hemoglobin. So this is RBC, and inside RBC we have hemoglobin. The basic function of hemoglobin is to carry oxygen. Okay, so the hemoglobin, it is divided into mainly two components. One is your heme and other is your globin. So I will draw a basic structure of the hemoglobin so you can understand. So here is a ring, and this ring is called as your protoporphyrin ring. And when you place iron inside of it, suppose inside protoporphyrin ring we are keeping iron, so what will happen? The newly formed compound is called as your heme, and to this heme if we are going to add a globin chain, then the entire compound is called as your hemoglobin. So once again I will repeat. So first we are going to take a protoporphyrin ring, and to this protoporphyrin ring we will add iron, then the compound is called as heme, and to this heme if we add a globin chain, then the entire compound is called as your hemoglobin. So what is meant by porphyria? The basic meaning of porphyria is defect in synthesis of your porphyrin ring. Okay, so basically what I am saying is there is defect in the porphyria biosynthesis. So what will happen if there is no ring present? If there is a defect in the porphyrin ring, obviously the hemoglobin will also be defective, right? Since there are defects in this ring, right? So that is the concept of porphyria. Porphyria is there is defect in porphyria biosynthesis. Okay, so we will see the basic structure of the protoporphyrin ring. Suppose this is a protoporphyrin ring. It basically consists of 4 pyrrole rings like this. Let me draw with a different color. So these are your pyrrole ring. Here is your pyrrole ring, and here is another pyrrole ring, and here is another pyrrole ring, and here is another pyrrole ring. So sorry for that. So there are nearly 4 pyrrole rings which will make your protoporphyrin ring, right? These 4 pyrrole ring, or we can simply called as tetrapyroles. So basically the heme we are going to make, the heme, I already told you the hemoglobin consists of heme. So the end product, which is the heme, usually consists of 4 pyrrole rings, or we can simply say a tetrapyrrole ring, and eventually the 4 pyrrole rings will fuse to form the 4 pyrrole rings fuse to form the protoporphyrin ring, right? Protoporphyrin ring. And to this protoporphyrin ring we will add iron, and when we add iron to it, it is called as your heme. So basically the structure of a pyrrole ring is like this. Now as you can see, the pyrrole ring is going to have a nitrogenous base. These nitrogenous base are very sticky. So this is a pyrrole ring, and this is a nitrogenous base which is very sticky. So obviously these nitrogenous bases can either bind to iron, which is seen in humans, and the compound is your heme. Normally these nitrogenous bases, suppose this is a nitrogenous base of a pyrrole ring, they can attach to magnesium also, and this type of suppose here is another nitrogenous base, and here is a pyrrole ring, nitrogenous base and a pyrrole ring, nitrogenous base and a pyrrole ring. This type of arrangement is mainly seen in plants where there is magnesium in the center, and the compound that is seen in plants is your chlorophyll. So in humans the middle compound is usually your iron, which leads to heme synthesis, and in plants the middle component is your magnesium, which is mainly seen in the chlorophyll. So these nitrogenous bases are very sticky, right? So these nitrogenous bases will stick with this magnesium in the chlorophyll. So that was about pyrrole rings. Now we are going to see how these pyrrole rings are getting synthesized. Okay, we have to see how the protoporphyrin ring is synthesized, or we can say porphyrin synthesis. How protoporphyrin ring synthesis happens, we will see in a stepwise manner. So basically the 2 amino acids are your glycine plus succinyl CoA, which are coming from your TCA cycle. Okay, succinyl CoA, and this succinyl CoA eventually by a lot of enzymes will get converted into a compound called as delta amino levulinate. So or we can simply call it as ALA. Now what is the enzyme here which is converting the glycine or succinyl CoA into aminolevulinate or ALA? The name is very simple. The answer is ALA synthase. Now this ALA synthase enzyme is mainly dependent on pyridoxal phosphate, which is coming from or a precursor of vitamin B6. So once again, glycine is converted into succinyl CoA. Glycine plus succinyl CoA will get converted into ALA. Okay, by the enzyme ALA synthase, which is vitamin B6 dependent. So the step is quite simple here, but in detail the glycine plus succinyl CoA by the enzyme ALA synthase. I will draw this with a different color. By the enzyme ALA synthase will first get converted into alpha amino and beta keto adipate. Okay, this is the first step actually, and eventually this will get converted into your delta amino levulinate, okay, which is delta ALA. So here also the enzyme is ALA synthase. Let me clear this one. The enzyme here is ALA synthase. Okay, so both these enzymes ALA synthase are dependent on vitamin B6, which is pyridoxal phosphate. Vitamin B6 is usually a precursor form of pyridoxal phosphate. So as you can see here, we are getting delta ALA, and this delta ALA usually gets converted into, I will draw it here clearly. So we got a compound called as your delta ALA, which is delta amino levulinate. Now from this delta amino levulinate, we are going to form a compound called as porphyrobilinogen. Okay, now what is this porphyrobilinogen? Porphyrobilinogen is a precursor of your pyrrole ring. It means this porphyrobilinogen is a pyrrole ring, like this. I already told you there will be pyrrole ring with nitrogenous base, but it is imperfect. So to this imperfect pyrrole ring, we have to make certain modifications, and eventually we can get your heme. Okay, so what is the enzyme here responsible for converting ALA into porphyrobilinogen? And here there is water gets kicked out during this reaction. It means there is a dehydration going to happen to ALA. So obviously the enzyme name is ALA dehydratase. The enzyme name is ALA dehydratase. Why ALA dehydratase? Because there is dehydration. Water is getting kicked out from delta ALA. So that's why there is a dehydration, ALA dehydratase. And this ALA dehydratase, it is a zinc metalloprotein. It means we need zinc in order to make this enzyme function. Okay, and not only that, if there are increased concentrations of lead in your body, or there is lead poisoning in body, this can inhibit your zinc metalloproteins, which is your ALA dehydratase. Okay, so not only that, I would like to say that ALA synthase, there are mainly two isotypes in your body, ALA1 and ALA2. ALA1 is ubiquitous. It means it is present in all your cells, but ALA2 is mainly seen in erythroid precursor cells. Okay, so this is only the first part. I will describe that second part in my next video, because I want to keep the video as short as possible. So as you can see here, I will just show you the diagram which is given in your biochemistry. So here is succinyl CoA and glycine in the presence of ALA synthase and by the presence of vitamin B6, which is pyridoxal phosphate, gets converted into alpha amino beta keto adipate, and in the presence of ALA synthase again, it gets converted into delta amino levulinate, which is ALA. And finally, the delta amino levulinate in the presence of ALA dehydratase, as you can see here, water is getting kicked out during this reaction and it is converted into porphobilinogen, which is the first precursor of your pyrrole ring. And not only that, this ALA dehydratase is mainly, it is zinc dependent, and in cases of high lead poisoning, this enzyme can be inhibited. Okay, I will talk about the remaining steps in my next video. Thank you for understanding.